Pharmaceutical Processing, in conjunction with INTERPHEX, presents INTERPHEX Live.
Let me introduce our esteemed panel. We have Hillel Cohen, Executive Director from my alma mater, Novartis/Sandoz, sorry, yeah, okay. Rick Morris, Senior VP from Pall Biotech. Mike Ultee, and he's the Principal at Ulteemit BioConsulting. Okay? So, normally the lead-off question is pretty simple. And then we've kind of jumbled things around a little bit to address some things that are more current. So anyway, the easiest way to do this is, so Rick, all right, how much progress have we made in the last year? I mean, we do this every year, so we kind of do a reality check.
Okay, can you hear me okay? Yeah, it's working. Okay, so over the last year, there's been a lot of progress in the three regions. I would say the most progress we've seen regards investment in facilities and number of approvals has come from Asia, then Europe, and then the US is still further behind than those three regions, but it is accelerating compared to what it was, and I'll let the other guys speak to that in more detail, but it's picking up some momentum. Also, regards to biosimilars, there has been an approval in Australia now for Biosana, so that was good to see, made on continuous and single-use technology, again, driving intensification, lowering costs, and costs are coming down as well for the biosimilars, so I think overall, a positive year.
My thoughts are very straightforward. I think biosimilars are now much better accepted in the public than they were several years ago. I'll point to two places. Well, the biggest piece is obviously getting the patients and the run-of-the-mill doctor to accept them. Scientists, I think we've come to appreciate the science behind it. Just a few weeks ago, the American College of Rheumatology came out with a white paper where they actually said that, and it was joint with the National Psoriasis Foundation, which is a dermatology group. I may have it, okay, okay. What it comes down to is that was a statement they said that, in their opinion, all biosimilars are now interchangeable. Now that's an amazing statement, because what they're saying now is that even if the FDA doesn't grant interchangeability designation, from their perspective, a pharmacist can substitute without needing to contact them as a prescriber. So that's actually a white paper that came out there. This follows the European Crohn's ECCO Association, where they also did a reverse. They had been very hesitant about biosimilars. Now they're very supportive of biosimilars. You have a group of between 30 and 40 stakeholders, including patients groups, pharmacy groups, physician groups, suppliers, insurance, coming together in a group called the Biosimilar Roundtable under the umbrella of the Biosimilars Forum, one of the trade groups. But the forum basically is just providing the umbrella. There you have 30 or 40 people working together to try and come out with a consensus education document. And just a few months ago, they came out with a two-pager of what they call basic facts about biosimilars, effectively endorsed by all of these groups. So these are several major advances we actually didn't have a year ago. And as Rick was saying, of course, we had 7 approvals last year, 18 total in the US. There are many more approvals coming in Europe. And interestingly enough, there's quite a few of the approvals happen to have been products that had gotten first round, that didn't get approved the first time. They got complete response letters. But the companies have been able to go back, do what's necessary to address the questions, and these products are now approved. So effectively, getting a rejection doesn't mean that you're out. It just means you have to address the questions. It also means, incidentally, a lot of these complete response letters came from products that actually were approved in Europe. And even though they were approved and marketed in Europe, the FDA still came back with questions. That also tells us that health authorities are really not quite harmonized yet about what their expectations are. And that's something I think that's going to be need to be forward to. The two big topics that we're talking about now are, can we have even more streamlined clinical studies because the analytics are as strong as they are? And then the other one is the concept of global reference product. We can show analytically that products are effectively identical from the reference product, but almost every country around the world requires that you have to go back and purchase reference material in their country and do studies. And scientifically, from our abilities, that doesn't make sense. So those are two new topics that came up this past year.
Can I just, so Mike, why don't you add in that, and then Hillel, I want to get back on that's complete response letters, so just getting a little more on it. Go
Let
ahead.
me just say I agree with what was just said from both my colleagues, but from one of the, thanks. Get close enough to the mic. One of the aspects that's still a big hurdle in the United States is not so much approval anymore. I think people are learning how to deal with the regulatory authorities and understanding, and the regulatory people are understanding what's possible and so forth. I think the biggest hurdle now is more commercial, where there's an intense opposition from those who have the innovator drug, and this is a legal fights and separate licensing agreements that delay entry to the market. So that's why there's so few actually on the market in the U.S., even though, what did you say, 18 approved, So I think that's sort of a business commercial hurdle. From the scientific standpoint and the regulatory standpoint, I think we're there. I think it's just, and there's some problems of the harmonization, but not complete, but I think we're moving close to that now. But in terms of actually getting them on the market and seeing biologic drug prices drop, I think that's going to take, to get through this business commercial block.
Gotcha. Go ahead.
Well, there are a couple things there. I think that we should have realized there are three major classes of barriers. The first is regulatory. Now, we've overcome most of those, although interchangeability, in my opinion, is still a barrier. Just take a look. Since the FDA came out with the guidance, nobody's actually doing the studies. People are voting with their dollars, and they're saying, we don't think these studies are feasible. So there's a regulatory component. A bigger component are patents, the patent estates. And it's interesting, this past month, AbbVie's been sued by 6 different groups, but patent estates are a big problem. It's not just every single biosimilar has had to go through litigation beyond BPCIA. And the third, as Mike's saying, is the commercial, rebates and other parameters. Reimbursement is another issue. Reimbursements haven't been resolved. So there's clearly are barriers out there well beyond the science.
Getting back to on those complete response letters, or the things that were contained in those relate to any of the issues that you just brought up. I mean,
I would say there are the quick hits and the longer, and there are a couple companies that got complete response letters, and we haven't heard from them since.
Okay.
Okay, that's one. All right, there are problems that.
Yeah, we get that. Okay, right.
There are other companies where they were asked, including my own. We filed today pegfilgrastim. We announced that we refiled in the U.S. To do that, we had to redo a clinical study. So there have been times when the FDA says, do this and this clinically, but then there are products where we said, the FDA told us do another clinical study, and we said, we're out. It doesn't make sense for us to be 4th and 5th in the markets. And so the last class of complete response letters have to do with really CMC issues, packaging lines. Pfizer's EPO did not get approved, even though there was nothing at all wrong with the product that I'm aware of. And I've spoken to several people about that, except the facility had gotten a warning letter. McPherson's facility in Kansas, until that got resolved to the satisfaction of the FDA, that product couldn't get approved. I think Celtrion had similar issues related to documentation that held it up. So there are some CMC documentation types that have held up some products as well.
Okay. One of the things we kind of huddled before the panel began, and what we thought we'd do was bring up the latest and the greatest to make sure we got that stuff up here. So one of the things that the late breaking news, well, not late breaking, but breaking news, would be we're going to talk about what happens when a biosimilar or interchangeable biologic, when the range of a quality attribute of the reference biologic changes after the biosimilar interchangeable biologic is approved. Got that? Okay? So we'll start with that one.
Well, the first question that they have along that line is, what does a manufacturing process change? What does the sponsor have to do when there's a manufacturing process change? This actually was a biologic user fee commitment to come up with a guidance about that. And the FDA very quietly did that. They sort of hid it in a guidance, draft guidance, a Q&A guidance issued in December. And it's very straightforward. They're basically saying the biosimilars has the same requirements as any other licensed biologic. That is to say, you have to follow ICH Q5E, and it's a long title. Comparability of biotechnology is subject to change in the manufacturing process. So when the FDA was very clear, when there's a change in here, you have to compare the biosimilar to itself when it was first approved. Because when it was first approved, that's when you established safety and efficacy. Agreed that it was based on the data from the reference product,
Right.
and you showed what the high similarity, et cetera. Now, the reference product has the same requirements. When you make a change to the process there for a reference product, they too have to go to ICH Q5E. But there is no requirement now in the guidances, and they're pretty clear about that the reference product has to go back and continually recheck itself in terms of biosimilarity with the reference product. So it is possible that there may be divergence over time in some quality attributes, but every time of the sponsor, whether it's a reference, the manufacturer of the reference product or the manufacturer of the biosimilar, they have to go back and compare themselves to their own products when the product was first approved. So you don't have to go chasing after changes. There are publications that have documented changes in the reference product over time, and there have been at least 2 cases that I'm aware of, both with my own company's products, where we met all high similarity to the reference product, let's say 3 years ago, but the newer, most recently released material had changed. And this was in a bioassay, no less. And we had to show that the change was not clinically relevant. But that will no longer be required as per the guidance that they have out there.
So to address something, if I had to make a process change post-approval, I mean, is that how this is going to be regulated? You just compare to itself?
It'll be as of like all biologics, subject to ICH Q5E. That's going to be the touchstone standard.
Gotcha.
So now they were very clear not to make any statements about interchangeability, because the interchangeability guidance is not coming out till the end of May. But I can't see how that would be different.
Well, yeah, I'm glad you clarified that, because I just assumed it was. Okay, great. All right. So we're going to kind of, anybody got any question on that specific aspect here? Because we're going to move on to another topic. We good? All right. All right, I thought somebody was yelling at me. All right, good. All right, so let's come around and, so we're talking about biosimilars. We ask this every year because it's incredible how many questions we get, and I apologize, Hillel. So why are biosimilars not generic? So Mike, you want to just clarify that again?
Okay, yeah, it's many people come from the small molecule industry where you are chemically synthesizing the drug. And when you're making it generic, it is an identical copy, an identical molecular structure to the reference or innovator product, the brand name drug. If you're making a biologic, it's done in a biological system, in a cell growing in bioreactors, and there's natural variability in the way the cells grow and so forth. And furthermore, these are much larger and complex molecules. These are typically 100,000 to 200,000 molecular weight. You know, a small molecule drug might be 500 molecular weight. So you see there's an enormous range, a big difference, big multiplier of 500 to 1,000x bigger. So it's not just the size, though, because it also has something called post-translational modifications, which are carbohydrate is the most famous of these. These are glycosylation added, and there's more variability possible there because post-translational means after the protein is made from the amino acids, other process steps take place where, in this case, carbohydrate is added on. And those can be branching structures and N-linked as well as O-linked. I won't go into all the biochemistry of this, but this produces a very complex structure such that any one particular approved biologic is not a pure substance. It's a family of closely related relatives. If you do any of these analytical tests, you'll see multiple bands on a licensed product that's considered pure in terms of biologic, has activity, and many of these changes, minor charge changes, even plus or minus some carbohydrate, may not have any effect or the safety or efficacy of the product, or even the pharmacokinetics, but it could. So you need to know what those process changes are, what the heterogeneity of your molecule is, and show how it compares to the reference product.
Yeah, we got a question.
Yes,
right?
sir. Yeah.
Yeah, we actually have done that. The issue become.
Hold on a second. Yeah, we didn't. Not everybody heard the question. Yeah, why don't you grab a?
So the idea would be to get ahead of this potential challenge by looking at possible patterns and post-translational modification ahead of any regulatory compliance comments, right? So trying to skate where the puck might be, so to speak, right?
The expectation of the health authorities is that there shouldn't be any new glycoforms, but there can be variability in levels of glycoforms. So if any company introduces a product that has a different glycoform, either if a biosimilar tries to get in it, that's going to be a challenge. And likewise, if the reference product tries to introduce a process change that brings in a new glycoform, they're going to be challenged with that as well. So it's fair as... Well, altered peptide ligands is another thing. Post-translational modification, I was talking about I was talking about carbohydrate structures. The other things that you have is you have amino acid substitutions, low levels of norleucine versus leucine as an example. I mean, the sequence codes for leucine, but depending on your cell culture, you may have substitutions of some variants.
Totally agree. One way to try and sort of make sure that that is happening as you make as well is you do more sensing, more quality control along the way, making sure it all ties back together. With the advent of more single-use, continuous, you try to tie that all back so you've got an intelligent system. That will all help in that as well.
Okay. We got the good. All right, I got a. My question is, and again, my job is to ask the dumb questions, so you guys can ask the good questions. So actually, I kind of interchangeable biologics, biosimilars. So one's better than the other. One's easier than. What's the deal on this? So
Yeah. Oh my gosh.
you're allowed to start. Yes, please.
These questions?
This actually, however, is become one of the biggest pieces of misinformation in the year 2018, 2019. What's happened is that the public at large, it's almost like a game of whack-a-mole. Now people accepting biosimilars. Well, wait till we get the interchangeable biosimilar. To get that designation, you have to provide additional clinical data. But the product itself is identical. You have a biosimilar, and you do additional studies. And the FDA has been very clear about this too. If you make a change to something so that it's you want to get into an interchangeable status, it's no longer the same product. Go back to start. The quality of an interchangeable biologic and a biosimilar, they are identical. It is the same product. The difference is you've gathered more clinical information to go switch back and forth. Now, I'll be a little heretical. From a scientific perspective, I don't see a difference between the products. They are the same product, for God's sake. It's a regulatory designation that's built into the law. And now that it's in the law, we have to find a way to adhere to the law. So the FDA came up with the concept of switching studies. But the product itself is absolutely identical. Having said that, it's easy for people to say, well, if you're getting more data, it must be a better product. But the reality is, it is the same product, but you've done an additional clinical study. Now consider, this is relevant, interchangeable is relevant, that the pharmacist can substitute without having to ask the doctor. What about products that are given in a doctor's office or in a hospital setting where the pharmacist is not involved? And we have, actually, most of the products, biosimilars today, are those products. For those products, there's no value to go and do an interchangeability study. And companies won't do it. There's no reason to do it. So it only refers to a product that is administered by a pharmacist. So you have the filgrastums, example, that are given in infusion clinics or EPOs. No one's going to come out with an interchangeable version of that because there's no reason to do that. So it's not a higher quality. It's just for certain products, it makes sense to go after that designation, but it's getting more clinical data. The quality is the same.
Mike, you got any? Thank you. Yep, there's one over. Yeah, Parrish. What?
Yeah, I'll yell it out. So one of the original concerns about interchangeability was traceability. In other words, if the pharmacist can switch, there was a concern about if there was a patient reaction, how would it be traced back? Which was it? Was it an innovator? Was it a biosimilar?
Well, the question of pharmacovigilance is really not a, this is my own opinion, but I think I echo a lot of people. The issue of tracing should be true for all biologics. It's not uniquely a biosimilar question. Different companies, different countries rather, have approached it differently. What seems to be gathering is that a pharmacist, well, the laws have to be changed to allow a pharmacist to do this because the laws are written for drugs, not biologics. But it's been changed in 45 states plus Puerto Rico. There are only 5 left. And for each of them, there's a requirement that the pharmacist must record the name and the lot number that's been dispensed. And if they make a switch, they have to inform the physician after the fact. And with any other switch, you have to inform the physician that I'm giving you this product instead of that product. So there is a requirement for the pharmacist in the interchangeability space to mark down what it is. Different states vary by the duration of time by which they have to keep the records. And there are a couple of more requirements. So there is a documentation requirement built into interchangeability. Now, this is independent of the question of the naming and the naming controversy. Whether it gets a suffix, whether it doesn't get a suffix, retroactive or not. But to directly answer at the interchangeability level, there is a legal requirement for documentation.
We good? All right. On this whole, I think one of the things that we always get into is people think it's an easy entry. You know, in other words, well, it's already made, then my process is going to be pretty easy. You know, how? So Rick, let me ask you this. I mean, I'm gonna make a biosimilar. So is it easier for me to do that than it is to start from scratch? And so
No, I would say the equal amount of effort goes into biosimilars as well. You obviously don't have maybe the same level of research to find the molecule. You're going to focus in very quickly.
Right.
So that reduces time and cost. But regards making it day in, day out on batch to batch or perfusion, you've got to be very diligent, have your traceability, your QC, QA already. If anything, at the end, you've almost got to provide extra support to the documentation to say that it is similar, which is, I think, an unwelcome burden that goes on the products, but with time I think that will change as well. And I think this is a good step that Hillel showed earlier, that you only chase the reference material you started with now, not one that keeps moving. As manufacturing processes get intensified, I think that will also improve the quality of a product because there's less hold points. You won't get as much aggregation, you won't get as much HCP, DNA, all that good stuff coming off that you want to get rid of. The product will be more consistent and batch to batch will be less variable.
I think compared to developing a new drug, Russ, as you said, I think overall the amount of work might be about the same, but your hurdles are different the amount of work in different areas. For a biosimilar, you have a huge amount of analytical work. You have to fully characterize not just your candidate product, but a number of lots of the reference drug, and typically at least like 20 or so. So you need to start collecting those and comparing them and find out what the bookends are of all the different parameters you can test. And these can be up to 50 different tests, because if you fully characterize, you're looking at details on glycosylation, other post-translational modifications. You're looking at degradative amino acids, for example, methionine oxidation or a terminal lysine on an antibody, other things. So they're sensitive amino acid side chains, and some of those will change in the course of production and over stability. It may have no effect at all on your product's efficacy, safety, or even PK, but you need to think about, well, how does this compare to the reference product? So what makes a biosimilar challenging is you're constant referencing to the reference product. You must constantly compare them side by side. If you're making a new product, you know, you're controlling the game. This is a new protein. You're producing it. You know, you need to fully characterize it, but you don't have to constantly compare against the current product. So where you save in a biosimilar, though, is in the clinical testing, because that's much abbreviated. And especially if you're going for multiple indications, you know, you don't have to test all those indications. You have to show efficacy in a primary indication, and then you can get multiple indications awarded to you. But you have to prove that you're highly similar, and not just somewhat similar, and certain things are given. You can't change the amino acid sequence. That's a set thing from the beginning of biosimilars. But all these other parts, you have to see, well, what happens if the glycosylation on my form of this is a little different? It might change the some of the effector function on antibody, for example, the antibody-dependent cellular cytotoxicity, ADCC. Well, you might make it better, actually, than the reference product, but then you don't only have a biosimilar.
Yeah.
You have a bio-better, and that's a whole other discussion. And that's a whole new product, and then you have to go back and do all that work and file as a new product. Now, is that worth it? It depends how much better your product is. If it's a lot better, you know, much lower dosage, fewer side effects, better survival rate of the patients, well, you can sell with that and has an improved product. But you can't use the biosimilar pathway to get your drug approved.
So it sounds to me, I mean, the challenges are on both sides, but to me, the challenges are more testing and analytical or process. Why don't we, I mean, which would you rate?
Well, let me just add one thing, because
Yeah.
these two gentlemen know more about process work than I do.
All
I'll
right.
grant you that. But in terms of testing, what the challenge you have in developing a biosimilar, one of the is that you have to develop the range for each of the critical quality attributes of the reference product. And if you only test 1 or 2 lots, you're going to have a very tight range. What you need to test it is actually many lots over time, and preferably material of different age. And you're limited by what you can buy in the open market. If you buy 5 lots and discover that of drug product, and discover they all came from the same lot of drug substance, well, it's a problem.
Yeah.
But we've seen changes in quality attributes of reference products over time, and that's perfectly fine. We're assuming that they've convinced health authorities that there's no problem, and we are happy to do that. People take this all the time. But you have you still have to get many lots over time, and that just takes time and costs money.
Right.
Agreed. And regards process versus analyticals, I think both go hand in hand. You can't almost do one without the other. Starting all the way from the media coming in to the bioreactor to further down, is you've got to have accurate data. If you can intensify, great, because it takes out hold steps and all the rest of it, but you've got to have that data on multiple lots, as Hillel said, but because otherwise you don't know what your upper control limits are, your QBD and all the rest of it around the process. You've got to get that in place and run it day in, day out.
I think throughout the process, the analytics are paramount for a biosimilar, and you have to make sure during your scale up and during your downstream and so forth, you don't fall out of biosimilarity. In other words, if something happened, your bioreactor went a little longer, or some other things changes, you might get acidic variants, you might get post-translational differences, you might have other problems that you no longer like the one that you made 2 lots ago. So now you have a new challenge, and unless you have good analytics, you won't know that. So you have to really push that.
Okay. Well, let me ask the question a different way then. So I'm going to make a biosimilar. So is there a specific process and method, analytical method I should be looking at that's going to alleviate some of the problems? You see what you get my question, right?
I think the essence is the FDA is pushing for what's called orthogonal methods. In other words, if you measure something, they want to see the same parameter measured by multiple different methods. If you're looking at size, they want to see 2 or 3 different size measurements. You're looking at amino acid sequence. they would like to see multiple ways. Having said that, to the best of my knowledge, there's no single assay that's paramount. And in fact, we've actually just published a chapter on that, showing that different companies address the same question to look at the same quality attributes by using different types of assays. So I don't think there's a single assay. Now, you have categories. You do peptide mapping, but no one's saying which enzyme you need to use for digestion. Something like that.
I would agree. I really see a lot of work in the bioreactor. A lot of analyticals there to make sure what you are starting off with is what you think it is. So there's a lot of work on glycosylation, the different sugars, everything that goes in. But again, you need to run multiple batches to build up the database and what your control limits are. And then you should be able to eventually run off standard readings saying that the process hasn't been perturbed, but you've got to run all the 20 lots or more to get there.
Okay. Mike, oh, we got it. Yes, sir.
So, attacking a little bit more of this post-translational modification, can you gentlemen comment on any RNA analyses maybe looking at as a way of controlling the amino acid sequences and then subsequently any post-translational modification potentials?
Well, the seed starts out the same, so that doesn't change. So the coding sequence is going to be the same. Okay. Well, then the question now becomes, well, the coding sequence itself should be the same. The translation can vary. Now, what you're looking there is looking at variants and impurities for each lot. And then you look for changes over time. Now the question is which post-translational modifications are most critical to look at. The FDA had a guidance on analytical similarity, which has been withdrawn. So yes, that was withdrawn. They don't withdraw guidances often, but they did withdraw this one. In that one, they had 3 tiers, saying the lowest tier was just look at and report the data. The second middle tier was stay within the range of the reference product, and the most stringent tier was do analytical statistics. And that's what got the whole guidance, that particular guidance, into trouble, because statisticians speak a different language, and I don't speak statistician. But what ended up happening is that the FDA said you should compare the means, and various sponsors gave reams of data showing the means sometimes can change, but the range doesn't. Or sometimes the range can widen enormously, and the mean stays the same. So the FDA acknowledged the validity of some of these questions about statistics, and they withdrew the guidance. Turns out it's a revised version is due May 31st. We'll see what they come up with, because it's actually a user fee guidance deadline. So they have to do it, or they have to report to Congress why they didn't. So they can miss it, but they're incentivized to make it. That's why I think it's more likely to be a draft than final. But the controls, to answer your question directly, once you identify what's the most important parameter, those that are most important are the ones that you have to control most tightly. As an example, there's an infliximab monoclonal antibody on the market, 2 of them, where there's a glycosylation pattern that's different than that of the reference products. And it turns out that that particular molecule, it doesn't impact the mechanism of action. So fine, that's okay. There are, however, glycosylation sites, carbohydrate sites, that if they would be changed on the same molecule would have an impact. And that one I was talking about was fucose levels. But for those that are critical, there's a very tight measurement. And quite honestly, if you're out of that, if your lot falls out, you can't use it.
I can add on the amino acid sequence you first asked about, that's fixed. But you have to, after your transfection, you have to select stable cell lines. And you may think you're stable cell line, but it could mutate over. So you have to do the standard, what you would do with a new product, 20 passages, 50 generations, to show that you got a stable cell line that doesn't, suddenly change its sequence. That's just part of developing any biopharmaceutical. It's not any different for biosimilars.
In the progress of our testing, though, so Hillel, let me get, because I think we opened the box, or something that confused me. So we track the reference, So, we look at it, we throw the best method we can at it. We think we've characterized all the CQAs, critical quality, right? And tomorrow we improve we do what's right. We improve our tools, right? And lo and behold, we find that there's something we didn't count on before. Now, are we stuck? What do we do? Are we back to square one? I mean, what's up?
That's actually happened, and it's been published. I'll talk about there's a molecule called etanercept, and that was where the analytics that we threw at it for the bioassay, I don't know if this is working or not.
It bounces in and out.
Okay.
But
The analytics showed that the potency actually dropped from the reference product over time. And this hadn't been something that was known before. However, in the clinic, there hadn't been any problem of loss of efficacy. So there have been a couple of examples. We since found out that the molecule refolded in the bloodstream into the proper conformation, because the problem was the drug product in the vial had misfolded proteins. Once injected, it refolded properly. So it turns out it wasn't a clinical problem. But there have been a couple of examples we've seen in analyzing biosimilars with surprises with the reference product that hadn't been known before. And health authorities are reluctant to pull a reference product off the molecule if there's no evidence of a problem. But it does create a problem. And it's going to happen more and more. In fact, the question when you see an analytical difference is what's the clinical significance of that?
Well, I mean, that's what it comes down to all the time. And I think it comes down to risk management. I mean, that's really what we do, a risk analysis, or do you go back in the clinic? What do you do?
I would say the tolerance for risk of a biosimilar is low. There's really, this is from a biosimilar manufacturer speaking here. There is a viable product out there called a reference product. If there is residual risk, there's no reason for that biosimilar, with a biosimilar that you cannot resolve. Remember, you cannot do clinical studies to address a question in analytics. If there's a question in the analytics, it's there. The clinical trials are not as good a tool as the analytics. That's
Agreed.
what we have. And it's a hard sell for physicians and patients. And the scientists who know that, but we have to try and educate.
Right. I actually have an interesting story about a homologous series I had worked on the small molecule side, but we won't get into that because the answer was it wasn't pretty, I mean, anyway. Yeah, I think, Mike, we talk about maintaining, we're in the biosimilars, our job is you track the reference, you maintain your process, your product. So we create this CMC thing that we put together. I think one of the things at least I fall apart on is in a biosimilar, how does that compare to a new? Is it the CMC the same, or is it like an ANDA and NDA? Are they the same, or what's in there?
I'll build on what I was saying earlier. At every step in your process, you're comparing to the reference product, and you're not doing that with a new product. Your new product, you make multiple lots and so forth, but you're not comparing against anything else because there's nothing else out there. It's a new program. So this need to match the biosimilarity. And what this means practically is when you're developing a new product, you may be selecting clones that are going to produce the very highest titer for cost efficiency and so forth. Instead of 1 gram per liter, you can go for 2 or 3 or whatever. And the standard now for antibodies is about 3 grams per liter. So some people get much more, some people less. But that's about where it is right now based on what Eric Langer presented this morning from his large survey of a lot of companies. When in biosimilar, you might have to reject those great producing clones because they're not producing a product that's similar enough to the reference product. You wouldn't have to do that if you were developing a new protein, because that's you're making the standard based on your high producer and so forth. Now, there are the other things I mentioned, the gentleman over there about cell line stability. Those are all the same. A lot of the process development and the manufacturing is the same, except for biosimilars, you've got to match the reference product. And so that's what makes this CMC especially challenging for that.
Right, gotcha.
And it's heavily based on analytics. I've seen diagrams, for example, that show a pyramid with the biggest thing on the pyramid. The bottom of the pyramid is the analytics. for biosimilars, and the smallest part of the pyramid is the clinical trials. It's just kind of like the opposite on other on a new product where the clinical trial, at least in expense cost, is way up there, you know, but you save a lot.
Right. So we're starting to run down on the time, but I think what we need to do, because one of the things that's always brought up is continuous. We're going to get to that, but in the meantime, what I'll ask Mike and Hillel to think about is, the areas of biosimilars. Where do you see in the next 12 to 24 months? Like a take-home message here. In the meantime, I'm going to ask Rick. So continuous. I mean, is there's a lot being said. I mean, is this relevant to this?
There is a large interest from biosimilars and also from CMOs towards single-use, intensification, hybrids, modular, continuous, because it's helping to shrink the footprint. If you can put more things together, there's less holds. The product is more similar, less variance from lot to lot. So it is gaining traction. Analyticals need to step up to match the same pace as what the individual processes are now providing. And then also with that data, a lot of data is now being generated, how you manage that and what you do with it, because you may not want to have everything that you measure in the PD level going all the way through to the manufacturing level. But there is definitely a large uptake into continuous and single use.
What I find interesting, you say it's the continuous seems to be nested in the CMO aspect rather than in the innovator aspect, or?
I would say, I have to be careful how I answer this.
Well, don't let me put you on the spot, but answer it anyway.
I would say there's a large take-up all around, but where cost is predominant, that's where there's more focus on hybrid and continuous.
Right.
Because it helps shrink the footprint down and the whole process.
Rick, how do you manage a situation where you're not really sure what the volume that you need for the market is?
That's a risk always. Some will be blockbusters, some will be personalized, but you scale the products as well. And we do have PD, GMP, and then we have large-scale GMP, and then the fluxes at the different levels are balanced. So we can do everything from like 3 liters an hour up to 350 liters an hour.
But are these continuous truly tunable? I mean, that's what I think it's important to understand.
You gotta mix and match, say, clarification to the inline concentrator to the moving bed chromatography. You have got to balance it out somewhat with a buffer in between. But generally, once you've got the main thing worked out, which is usually the chrome step, everything else around it can be matched.
Okay.
I would like to add that going continuous or some other highly efficient manufacturing technology is not going to help you in terms of biosimilarity, because the innovator reference proteins were probably not made that way because they didn't exist. So what you are gaining is cost efficiency, and as biosimilars mature, costs will become much more important. So I see the future there in terms of getting costs down, and you get a competitive landscape, which we don't really have now, at least in the U.S., but we will as more the biosimilars get on the market. Then there's going to be cost sensitivity around the production of these very expensive drugs, and I think that's where going to efficient processes with a smaller footprint that can run continuously, that can become an advantage. But in terms of making something similar to the reference protein, no, it's probably not going to be an advantage there.
I think we're also going to see a shakeout in the industry in the next 24 months. This past year, we now have 5 companies that have dropped out completely, that I'm aware of, one of which dropped out this past year. More importantly, and more concern to me, is the fact that there are some major players that have decided to not start programs or discontinue existing programs. And the attitude of these companies is that we want to see that we're already starting, that it's a profitable business. We don't want to invest more. So I'm not sure if it's going to shake out that the large companies will be able to do it and Joe Sunoco and Drug Emporium will drop out. Or it's a situation where the companies in India and China with lower cost of goods, or any company with lower cost of goods, we could do it, and the large pharma companies saying, you know what, we're better off spending our money elsewhere because we're not getting a good enough return. But I do expect that we'll see some shakeout in the industry and the players in the next 24 months.
Right, and that shakeout should be an uptick in the guys going after the biosimilars, or
Not necessarily. I just did an analysis, and comparing 2018, a year ago to today, we have almost the exact same number of biosimilars in development. I think my calculations are 4 more by the FDA numbers, but you can't really tell because we know that there have been biosimilars that have been discontinued programs, and we know there are new ones. So net, we're around the same in terms of products in the pipeline in the U.S. There are more products under review now than before, but that's just a function of where we are. The real question is going to be, is the pipeline filling up at the early stages? And there's reasons to be concerned about that.
right?
One other comment is the amount of investment is over $1 billion into plant in China for biosimilars. For Europe, it's probably half of that. And then the US, as you guys just mentioned, that seems to be holding steady or declining. So where will the biosimilars come from based on that plant expansions, or what's going to come from Europe and China?
Well, I want to thank our panel members, Mike, Hillel, Rick. Thank you very much. Thank you.